Hello everyone. So this is going to be the last screencast actually of this semester. So what we're going to discuss in this screencast is how you can quantify DNA and how you're going to be quantifying it in this lab this week. So that's the very end of your 4-week experiments where you have grown your bacteria, you harvest them a week later, then you extracted the DNA last week. Some of you got awesome pellets, and so now it's time to figure out what is the mass of DNA that you actually got out of these 4 weeks experiment. So in general, there are two ways that you can go when it comes to quantify DNA. The first way is by using UV spectrophotometry. This is the fastest method. That's very commonly used in the lab. And the advantage of this method is that DNA is read as is. So you just take an aliquot of your sample and you read the absorbance at 260 nanometers. So DNA doesn't need to be modified in any ways or to react with anything. The other advantage is that it allows you also to check the purity of the sample, and more specifically to see how much protein you have in percentage in your total sample. The drawback is that you need a UV spectrophotometer, which is more expensive than a regular spectrophotometer. So this is a spectrophotometer that allows you to measure absorbance at wave and low wavelength, so in the UV range. It is very different from a regular spec 20. The second method, and that's the one you'll be using in the lab, that's the only one we'll be doing in the lab this week, is by generating a standard curve by getting DNA to react with a labeling reagent, and then reading the sample with a very basic spectrophotometer like the spec 20. So the advantage of this method is that it doesn't require any expensive equipment or type of spectrophotometer. The drawback is that you have to carry on a reaction, which obviously takes longer than reading the sample as is. So let's focus on the first method, so how to quantify DNA by spectrophotometry. So DNA, as you can see in this absorbance spectrum here on the bottom, DNA, if you do an absorbance spectrum here in the UV range, you'll see that DNA absorbs very, very well at 260 nanometers. So if you take a sample of DNA, you put on the spec 20, you can actually use then this wavelength, 260 nanometers, to do the absorbance. In this case, since you're working with your ultraviolet light, you actually need special cuvettes, which are quite expensive, which are made of quartz. Now, how do you quantify DNA? Well, we know that if you take a sample, 50 micrograms per milliliter sample of DNA at pH 7, and you use a 1 centimeter cuvette, now you read it at 260 nanometers, you get actually an absorbance value of 1. So now, if you take another sample at this point, and this gives you an absorbance at 260 of 0.86, it's actually very easy to determine the concentration of the sample by simply doing a ratio, which is nothing else than the reasoning, which is if 50 micrograms per milliliter of DNA gives you an absorbance value of 1, what concentration, obviously micrograms per milliliter, will give you an absorbance of 0.86. So the concentration at this point will be equal to 50 micrograms per milliliter times 0.86, obviously divided by 1. We could omit this part. So it would be proportionate to this. So that's a very simple way of determining the concentration. You just took a sample of your DNA, you dilute it, you read it to 160 nanometers, and then using this proportion here, you can figure out, based on the absorbance you get, what is the concentration. As I said earlier on, the advantage of this method is that it's fast and also allow you to figure out how much protein you're carrying over from the previous steps. If you remember last week, you got your DNA out in the aqueous solution on the top after you centrifuge with chloroform and isoamyl alcohol. but it's very easy to carry over some SDS with some protein. So you want to know how much protein you're actually carrying over. And so you can do that by actually measuring the absorbance not only at 260 nanometers, but also at 280 nanometers, because at this wavelength, proteins actually absorb very well. So now, if you can actually figure out the absorbance at these two values, you can calculate a ratio between the absorbance at 260 and 280 nanometers. And this ratio will give you the information on what is the percentage of protein which is contaminated in the sample. Here on the right side, you see this table tells you how, based on the ratio, you can determine how much in percentage is the protein. So for example, if you have a ratio of 1.79, you know that you have roughly 62% of protein contaminated in your sample. So I mean 62% is protein, the remaining 38% is DNA. So very little DNA, lots of protein, not a good sample to keep for your experiment. So in general, then when you use this method, what you're aiming at is a ratio which is at least 1.9, which means that if you look here, the percentage of protein contaminating is above this, so it's actually less than 1% protein contamination. So your sample will be very pure or quite pure. It could be used for further analysis. All right, so together with this method, now there is a second method, which is the one you'll be using in the lab, and is the one of quantifying DNA by performing a reaction, labeling reaction with this chemical called diphenylamine, or DPA. Now, DPA reacts, if you put it at high heat, it reacts with 2-deoxypentoses sugars bound to purines. If you remember, DNA contains 2-deoxyribose, which is a 2-deoxypentose. But it reacts only DPA, only to, in this case, 2-deoxypentose bound only to purines. It means that if you take your DNA and you get it to react with this chemical, only half of the nucleotides will react with DPA. Okay, this is very important. We'll see later on when it comes to do the analysis of your data, how you have to take this into account. Now, when this chemical reacts with DNA, what you get is actually a blue color, which then can be quantified using a very basic SPEC20. And now, the amount of blue color that you have produced here is actually proportional to the amount of DNA. So, if you can generate a standard curve with known masses of DNA or a substitute, then you can use that standard curve to figure out how much DNA you have in there. So, as we say, so first step when you quantify DNA using DPA or diphenylamine, first you generate a standard curve with known masses of DNA or a substitute of DNA, which is like deoxyribonucleotides, contain a purine, so which is 2-deoxyadenosine in your experiment. So instead of using DNA, which is expensive, you just use a deoxyribonucleoside, which is 2-deoxyadenosine. Notice this contains a purine and therefore will react with DPA, diphenylamine. So now, once you've done this, you can actually take 2 samples from your own genomic DNA that you have isolated in these 4 weeks, and then you can measure the absorbance, do the same reaction, measure the absorbance, and using the standard curve you generated with known masses of deoxyadenosine here, you can actually figure out how much you have in there. So how do you do it? More practically speaking, so you'll have to set up a series of tubes where you use different volumes of deoxyadenosine here. Obviously, you would know the mass of deoxyadenosine, so you can figure out the mass that you're actually adding in each of these samples if you add these volumes right here. Then you bring all your samples to 1 milliliter by adding distilled water, and then you combine it with the reagent. Okay, so DPA, diphenylamine, by adding 2 milliliters. So these will be your standards. So once you get this to react for 10 minutes in boiling water, now you can then measure the absorbance of this sample and generate the standard curve you see right here. So first question I have for you, first of all, how do you figure out the best wavelength for these samples? All of this information is in your procedure. So I will not discuss it here. Read the procedure and you figure out how to do that. Second question I have for you, and again, I will not give you the answer, but I get you started on thinking about that, is which sample will be the blank in this case. Think about the definition of blank, which we went through several times throughout the semester, and do that. So now, together with this, when you do your experiment, then when you prepare your standard curve, at the same time, you also take two samples of your genomic DNA, so 0.5 milliliters and 1 milliliter, and obviously you bring them to 1 milliliter in total with distilled water, and you do the same reaction with 2 milliliters of diphenylamine, and then you can compare these absorbance to the one you have generated with the standard samples and therefore the standard curve, and figure out the concentrations. So here it's exactly what we're saying. I just kept it separate just to focus, but all of these samples should be done at the same time. So all of this reaction, okay, to save time. Now, obviously once you have generated your standard curve with the samples here in green, now you can use the absorbance that you get from your two samples. For example, if you get this value right here, use the standard curve as long as this value falls in the linear part of the curve and figure out the mass that you get. So which will be X micrograms of DNA which you had in 0.5 milliliter or 1 milliliter. Now, is this really the mass of DNA that you have in there? Well, think about something. You're not there yet. Now, I'll give you an example here. Imagine you have one sample of deoxyadenosine here, which contains 5 molecules. Sample number 1. And now, together with that, you have a second sample, which contains 1 molecule. I know it's a ridiculous example, but 1 molecule of DNA, which has a 5 base pairs. So in total, 10 nucleotides here, complementary to each other. Now, you get it to react with diphenylamine, both of them. The first question I have for you is, would you think that you would get the same absorbance out of both samples? Now, if you think DPA, we say it reacts only with purine containing nucleotides, so it will react with all of these molecules here, right? 5 of them. Now, what about in these samples? Well, the purine is containing adenine, purine, guanine, purine. So now you would have 5 nucleotides here reacting too. So you will actually have the same. Should give you the same absorbance, this value, despite the fact that here you have 5 nucleotides or nucleosides, and here you have 10 of them. So the fact that you get a same absorbance here doesn't mean that you have the same mass of deoxyadenosine and DNA there. Clearly not, because here you have 5 nucleosides and here you have 10 nucleosides, monophosphate, interacting with each other. So all of this, the point here is that to tell you that now the absorbance, the same absorbance that before on the slide before here, would have given this mass, doesn't really give you a mass which corresponds immediately to the mass of the DNA you have in there. In fact, it's actually underestimating the mass for 2 reasons. Now let's go through them and I'll show you also how you can account for them. As I said before, so DPA reacts only with 2 deoxyribose bound to purines. So only half of the nucleotide of the DNA sample will be labeled. Please keep in mind one thing I forgot to tell you earlier on, is that in reality DPA does not react with DNA, intact DNA like this, but the nucleotides actually get hydrolyzed. But in any case, so in DNA, 2-deoxyribose reacts only with half of these nucleosides. So in order to account for that, now to make the masses comparable, now the estimated mass of the DNA that you get from the standard curve has to be multiplied by a factor of 2, which accounts for exactly what we just say. Second thing that you have to keep in mind is that now you're trying to figure out the mass in micrograms. But now you're comparing the mass of these deoxyribonucleoside, deoxyadenosine, to the one of the deoxyribonucleosides, monophosphate, which are present in DNA, but their mass is actually different. If you look, this is basically the same nucleosides as you use it to generate the standard curve, and this is as is found in the DNA. In the DNA is found as a nucleosides, but monophosphate, so it contains also an additional phosphate group, which obviously affects the molar mass. So the molar mass of a nucleoside in the DNA is higher than the one of deoxyadenosine in general. And obviously there is also a molecule of water here that is lost during the condensation reactions of the different nucleosides in the DNA. So now, in fact, if you look at the mass of deoxyadenosine and you take the average of all the deoxynucleosides, monophosphate, which are found in DNA, there is a very significant difference here. And so, in the end, the mass that you get out of the standard curve not only has to be multiplied by 2, but it has to be adjusted by this difference, by multiplying by a factor which is nothing else than the ratio between the average mass of the deoxyribonucleoside monophosphate found in DNA divided by the mass of the deoxyadenosine right here. So this factor 1.228. Now, let's just give you an example going back to summarize what we just said and finish it up. Let's assume that your sample, one of the two samples, the 0.5 milliliters or the 1 milliliter sample, gives you an absorbance of 0.52. So now you use your standard curve and you figure out that this is the mass, 7.5 micrograms. But as we said before, this is not really the mass of DNA you have in there, because basically you're comparing DNA, which contains both purines and pyrimidines and has nucleosides, monophosphate, which have a different molecular weight than deoxyadenosine, to deoxyadenosine. And so what you have to do, so if that's your mass, you have first to multiply this mass by 2 to account for the fact that only half of the nucleotides will react in DNA. And then you multiply by this factor, which adjusts by the difference in molar mass of the nucleoside monophosphate in DNA versus deoxyadenosine. And this will give you the actual mass that you had, the correct mass that you had in either 0.5 or 1 milliliter of your sample. Now, the next step, and I leave it up to you, you've done this calculation several times. How do you go from this mass to actual concentration of your sample? You know how much you add there, and then you know the total volume you have of DNA, which you have suspended last week. And so you can do your calculations based on that. All right, that's it for this screencast, and then we're done with our semester. Bye, guys.